Our next speaker is Professor Hoitinga from the Netherlands. Thank you very much.
Thank you very much for inviting me here to speak. It's a great pleasure. And thank you especially to Lida Anestidu, whom I've known for many years and been cooperating with for many years. The title of my talk is Systematic Reviews, Meta-Analysis and Reproducibility, and Professor Deernagel already gave a very good introduction to that. My professorship has been changed name to Evidence-Based Laboratory Animal Science. It used to be Laboratory Animal Science only, but because I have made the move to focus on systematic review, the evidence-based has been attached to that. I founded SYRCLE, which stands for Systematic Review Center for Laboratory Animal Experimentation, and I will tell you more about that later. So this ILAR roundtable is focusing on advanced disease modeling in animal-based research in support of precision medicine. I think before advancing disease modeling in animal-based research, I think it's very good to reflect on what the current situation is around animal modeling of human disease. We are currently talking about a reproducibility crisis when we talk about animal studies and preclinical studies. A paper by Friedman has looked into the literature in 2015 how the reproducibility, or rather the non-reproducibility of preclinical animal and in vitro studies shows. And the non-reproducibility varied from 51% in the paper by Glacio to 89% in a paper by Bagley and Ellis. And what is striking is that the pharma first wants to reproduce studies that have been published to find out whether that is really also what they are going to find. So next to the reproducibility crisis, I think we can also talk about a translation crisis. This is based on two examples I have from stroke from the Camerades, where they have seen that when you have about 1,000 positive results from new treatments in animals, then only about 4 or even less, as Ulrich Deernagel just said, work in the clinic. We've also seen in previous presentation that the number of drugs that have been approved by the Food and Drug Administration have declined since 2000. And there is a publication by Los Calzo from 2012 which says that it seems like the big focus nowadays on target-based is less successful than what we did in the old days to focus on phenotype-based screening. Prince, who was one of the literature that was refereed in the Friedman paper, has mentioned the possible causes for low reproducibility, which are lack of reporting of details, lack of standardization, incorrect statistical analysis, insufficient sample sizes, interactions with different environmental influences, selective reporting of results, bias towards publishing positive results, significant chasing, and errors that remain undetected in our current peer review system. So, what to do? We have, in my department, first focused on the 3 R's, replacement, reduction, and refinement. And I've taken this PhD with me. You're welcome to ask me for a copy. I've only carried only one, because it's too heavy to carry more. But it's called The Next Steps Towards Responsible Animal-Based Research, where we have looked at how the 3 R's are dealt with through questionnaires, and then we have moved on to the systematic reviews afterwards, as we think that is more effective for implementing the 3 R's and doing much more with animals. So systematic reviews are up for the job, we think, for creating transparency on quality of animal studies and translation, And especially creating transparency will hopefully trigger improvements that at least has happened in the medical field. Our group SYRCLE is shown here, and the activities we do within SYRCLE is to provide teaching, because systematic reviews is a new methodology for animal studies, so you need to teach people the steps to take. We also coach people, so once they have been on a one-day workshop, we provide coaching so people can take all the steps and don't get stopped during the process, but we can help them continue. We've developed a free e-learning module. It is a leaflet on the table at the registration desk at the back side. You can see exactly how you can approach this free e-learning module. It takes about 1 hour to go through all the steps. We execute systematic reviews, and you see here the group that is in a project where we look at translation of animal studies of cystic fibrosis and rheumatoid arthritis, where we try to identify success factors for translation. We are also involving patients in the research process. And a hypothesis that has arisen from this project is that systematic reviews may be a good model for translation. We're also developing guidelines and tools to promote the process, and we're involved in international networks. We've also started an international network ourselves, which is the SYRCLE Ambassador Network of people that are promoting education of systematic reviews of animal studies locally at their places. And we are very happy. We have a lady from the Dutch health funding organization, which is called ZonMW in Dutch, who has helped us to make this all possible. Without that help, it would have not been possible, because there is not a lot of funding available for this type of research. In 2017, we got the second prize, which is called the Cochrane Reward Prize. Cochrane is the equivalent organization doing systematic reviews for clinical trials since 1992 already. And the Reward Alliance campaign was started by The Lancet in 2014 in order to increase value and reduce waste in research. And so it was for us a big recognition to get this prize, because as an animal field community, we want to engage in the medical field, because that's why we do most animal studies. So to be incorporated in that community, we hope to make real progress. So what are the steps in a systematic review? Most of you probably know that from the Cochrane. But it is very important to do these steps and do them as objectively as possible in order to reach evidence-based conclusions. So phrase research questions, search for all evidence, select relevant studies, extract the characteristics, assess the study quality, and if possible, do a meta-analysis of all the animal studies together. And that step is usually appreciated most by everybody, because that step creates the overview. What are the benefits of systematic reviews? We have seen that you can make a more evidence-based choice of animal models. For example, in the field of tissue engineering, of cartilage defects, it is very popular to use rodents. But in rodents, the defects will always cure, whereas the defects in the cartilage of humans will not always cure. And so, for that purpose, you need to use large animal models. Systematic reviews lead to more translational transparency, and I've shown you the examples of stroke and multiple sclerosis. It leads to the implementation of the 3 Rs, at least it can help to immediately. We did a systematic review with the surgery department, and it was shown that there was a lot of unnecessary repetition to demonstrate that chemotherapy reduces wound healing. You won't have to repeat it anymore. That is a known effect. And then last but not least, systematic reviews make reporting quality transparent. This quality assessment of reporting is one of the steps in a systematic review. For example, you look at whether publications report randomization or blinding. These are the two starting points of our scientific practice. But if we look in the literature, we've done thousands of articles now in systematic reviews, then 50 to 80% of the publications do not mention whether randomization and blinding have been performed. On the top, you see a very typical graph of reporting of quality indicators. It's very red. That means that it's not reported. If it's not reported, then you don't know whether you can reliably interpret these findings. So then we talk about unclear risks of bias, and that is illustrated by a big orange field, which we see again and again. The Camarades group has demonstrated that if you don't randomize and don't blind, you get two positive results. That's exactly why we do randomization and blinding, because of course you want your hypothesis to be true. So that's why you need to do randomization and blinding in order to get your results as objectively as possible. But getting a positive result may lead to a very nice publication in a good journal, but it can also lead to exposing patients unnecessarily. And examples exist in practice where patients were exposed on the basis of results of individual animal studies, where the clinical trial did not go well, and then people went back to the animal studies and did systematic reviews, and then it shows it could have been predicted. So low quality reporting is a synonym of low reproducibility. What I also would like to stress here, that a high impact factor journal does not guarantee a higher quality reporting and thus not better reproducibility. This is a paper by MacLeod in PLOS Biology 2015, where he has explicitly been looking for the higher impact factor journals in quality, and there was no correlation. Then how about reporting of animal studies? What about reporting guidelines? In 2010, the ARRIVE guidelines were published, Animal Research Reporting in Vivo Experiments. In 2014, the guidelines were adopted by 400 journals, which meant they were included in the instructions to authors. Baker did an analysis in 2014, which meant that the ARRIVE guidelines had not been implemented yet. Now, this year, the ARRIVE has been adopted by 1,000 journals, and we still see hardly any improvement in publication quality of animal studies. So, what to do? Last week, we had a roundtable in Edinburgh because we thought something needs to be done in order to really get quality improvements. So we invited funders, editors, researchers, and equator people to get together and talk about improving the uptake of reporting guidelines. This will probably need a culture change, and I think the main outcome was that probably funders and editors need to enforce ARRIVE, and we need to educate and train the next generation scientists much better. So a lot of commitment from funders, editors, and academia will be needed. And also it was stressed that laboratory animal vets should be involved much more in this whole process. Science magazine also commented on the low progress in following ARRIVE guidelines under the title "Sloppy Reporting on Animal Studies Proves Hard to Change." Also, the Camarades has done an intervention with the ARRIVE checklist, and also filling out the checklist before writing or sending in the publications doesn't help to improve. So it's obviously very complex to get this in. So I think the transparency by systematic reviews leads to subsequent improvements in quality of reporting. This at least has been experienced by the Cochrane collaboration, who have published a lot of systematic reviews of clinical studies where they had to publish the shortcomings of primary studies, which then led to an improvement of the quality of the primary studies. And it seems like history needs to repeat itself for animal studies. The Communal publishes a lot of systematic reviews on stroke, and I understood from Malcolm MacLeod that the Stroke Journal has a relatively high incidence of randomization and blinding now in their publications. If we would get a high-quality reporting, we will be able to do a more reliable assessment of translation of animal studies to humans, because at the moment we don't know whether a low translation is the result of the low quality of reporting, or if it is an effect in general that animal studies have a low translation. So when I prepared for this lecture, I asked myself, for precision medicine translation, what would we need? I think, first of all, make the choice of animal models more evidence-based, or what was at the latest conference, World Conference on Alternatives to Animal Studies, decided that we should have more human-based models. Then, for animal studies, a better quality reporting is necessary. And then, my dream is that future systematic reviews and meta-analysis encompass both high-quality human and animal individual data. That way, you can do a translational meta-analysis, and you can demonstrate by statistics what factors are essential to achieve precision medicine in humans. So, thank you very much for your attention, and if you have any questions, please use my email address. Thank you very much.
And we expect to have some time for questions at the end as well. Thanks very much. And our next speaker is Dr. Monroy from Harvard.
Hi everyone, my name is Arjun Manray. I'm a researcher at the Department of Biomedical Informatics at Harvard Medical School. Delighted to be speaking today about reproducibility in the context of biomedical computing and large databases. Many of the examples will come from human genetics. That's what we spend a lot of time working on, as well as the CDC and Haynes dataset, but I think many of the lessons apply to the topics that we've been discussing today. First, I just wanted to thank the organizers for inviting me. So I don't think I need to motivate reproducibility to this crowd, but I thought this was a good way to start. This is the great 20th century philosopher of science, Karl Popper, who said very directly that non-reproducible single occurrences are of no significance to science. So reproducibility is, of course, very old as a concept, but it's been all over the news lately. It's been very prominent in drug development. We've heard some of that from the previous speakers. But really, in biomedical computing, this affects the full translational spectrum from basic science to clinical trials. There's even quite a popular blog called Retraction Watch that follows retractions in science. And there's been, quite a bit of debate over numbers like the 6 of 53 from the Amgen study that looked at previous landmark cancer papers. And this has led, to wonder about the reproducibility of these reproducibility studies themselves. This, of course, may actually strengthen the conclusions of the original studies while potentially casting doubt on their methods. But either way, I think there's widespread consensus, as we can see in this meeting, that this is an important topic to address and to come to terms with. There's been many solutions, as you've heard, to help bolster or improve the reproducibility in biomedicine and animal science. I think the top thing that I see is individual responsibility. So this is better training in statistics. This was a study that was published in Nature. It was a survey of 1,500 scientists last year, and you can see that the top two factors are better understanding of statistics, better mentoring, supervision. Many of those other factors, I think, are individual investigator responsibility. So the question we have generally is, what about structural factors? What if there was no bias? What if there was perfect essential investigation itself by the investigator? What factors outside of the individual investigator's control would shape the reproducibility of their conclusions? And so where I think this is really, really fundamentally important is in the context of multiplicity. And so this was the initial observation that prompted the results that I'm going to discuss. These are two scenarios, and the one on the left is just a SNP chip. It might have a couple 100,000 SNPs on it that we measure in a GWAS or something like that. And we almost always address multiplicity explicitly and rigorously in that context. And if we have a large shared database, which I think is very common now, we've seen many examples of that today. In human genetics, it's also very common. There might be, in a process that spans decades, that we see many investigators analyzing this shared database together, and we almost never explicitly address multiplicity in that context. But of course, this process on the right here can resemble high-throughput genomics in scale, but not in synchrony. And if we're not very careful about how we address multiplicity in this context, we can end up in very poor reproducibility situations. So how can we study this problem? So when I was in grad school, I saw this paper. It was very inspirational. I'm sure many of you have read it as well. It's called Why Most Published Research Findings Are False by loannidis. And you can see that it's been extremely influential. There's over 2 million views on this PLOS Medicine article. I'm responsible for about 1 million of those. What it does is it gives us a framework and a language and a way to think about the reproducibility of a given scientific field in terms of this concept of PPV, which is very familiar from the diagnostic testing world, the positive predictive value of science, not of a given test, as a function of what is happening in the scientific community. And so the key parameters here, I've just highlighted two. There are many others in John's framework. R, which is the pre-study odds. It's the ratio of non-null to null relationships that are being studied in the field. And U, which is a bias term, which reflects the proportion of findings that are published that wouldn't be published if studies had been conducted perfectly to begin with. So our goal was to extend John's framework, and so this is work with John. I was very fortunate to connect with him in grad school. The goal is to extend this framework. So many of these parameters in his original framework were scalars or constants to these communal science situations in which many investigators are conducting these studies with studies of varying quality. And so in the setting of communal analysis of a shared data set, we have to generalize this model in order to parameterize reproducibility. So there's N and B. These are two big terms. I'm not going to dwell on the math. I'm just going to give some intuition here. There might be relationships being studied by only a small set of teams, or there might be certain relationships that have 100 teams studying each of those relationships in the given data set. And then, of course, as we've seen also, each study in this given analysis might have very different powers. So there might be some studies with 80% power, very rigorously enforced, and others with 10%, 20%, 30% power. And so then the last part of this is recognition and sort of explicit accounting for the fact that this quantity that we're defining, dPPV, the data set positive predictive value, is a stochastic process. It's a random variable itself, and it has variance even in these any of these settings that we're looking at. So the distribution that's sort of central to this quantity it's indicated by PB there. It's a Poisson binomial distribution. It's the generalization of the binomial distribution for non-equal probability of success on each trial. And this is a ratio distribution. It's hard to manipulate. We can do Taylor approximations and compute the expectation and the variance of this quantity and do a lot of analysis this way. So a lot of our work has gone into making this quantity analytically tractable. But where I think it's more interesting is thinking about it in the simulation setup that we've been working on. And so these communal science settings are ubiquitous. There are a few examples shown here. The primary example I'll talk about today is NHANES. This is the CDC data set, National Health and Nutrition Examination Survey, but we've seen very similar results with large-scale genomics data sets, for example, the Genome Aggregation Database and the Mouse Phenome Database as well. And so I've been discussing theory, but briefly to motivate this a little bit more, this is all in service of medicine. In the context of human genetics, we published a paper last year where we found, to our surprise, that multiple variants had been misclassified, and these variants were much more common in African Americans. So when we looked at patient records at a leading testing lab where individuals had misclassifications, they were much more disproportional in African Americans than other populations. So this type of irreproducibility is what we're trying to avoid with these frameworks. So this is our computational framework for studying communal science and measuring this quantity dPPV, the dataset positive predictive value. We start with a shared data set, and so this is N. Haynes for us typically in these analyses. And then essentially we use empirical distributions of that data set to simulate artificial variables that follow the same distributions so that we can look at pairwise correlations and control the fraction that are considered null or non-null over time. And what we do is we simulate different teams gaining access to this data set iteratively in time steps. Performing studies with different type 1 and type 2 errors. For example, in the context of N. Haynes, this is looking at the relationship between albumin and total calcium. Bias is used to select which findings are published, and then because we seeded this process, and this is all in a simulation framework for this data set, we can measure this quantity, the data set positive predictive value, over time. So this is the simplest possible scenario here, but I think it really illustrates the central point about multiplicity. So suppose there are 1,000 relationships that are being studied in a given shared data set. There's one study being conducted per relationship, and a relationship is, for example, the correlation between albumin and creatinine, or albumin and calcium, whatever we're interested in. There's the customary significance threshold of 0.05, and the pre-study odds, which is the ratio of non-null to null findings, is 0.001. And we have good power. Nominally it might seem that we're well set up here to do rigorous science, but if only the studies that survive that 0.05 threshold are reported, in expectation, the reproducibility of the claimed findings from this data set is less than 2%. And so the reason is that the power of our study, which is 0.8 or 80%, exceeds the false positive rate by a factor of 16, but the null relationships dwarf the non-null relationships by a factor of 1,000. So we're essentially doomed here. So that's the simplest possible scenario. The way a data set is analyzed in practice, I think, is well captured in this article by Gelman and Loken in 2013, where they invoke the Borges story, the Garden of Forking Paths. So essentially, you can chop up a variable, you can binarize it however you want, you can adaptively set your analysis after you see the data, and this is not necessarily p-hacking. It could be p-hacking, which many of you, I'm sure, are familiar with, but this could be, I think, very innocent sort of post-data inspection corroboration of your hunch hypothesis, which I think, I guess, broadly would be confirmation bias. But really, the way the data set can be analyzed can be very, very complicated. And this article does a good job of giving practical examples of that. So in the context of N. Haynes and looking at the shared data set, what hurts reproducibility is when teams arriving late, for example, tend to essentially pursue a priori less likely hypotheses. And so the key parameter here is R, which is the pre-study odds, the ratio of non-null to null hypotheses. It's shown in red here on the figure. And essentially, if this declines over time, then the dPPV, or the dataset positive predictive value, will as well. And so, for example, most a priori likely relationships might have been claimed by the early investigators who first had access to the dataset. And as the dataset was opened up to the broader community to essentially pursue novel hypotheses, what you'll oftentimes see is that a priori less likely hypotheses are pursued. And this is in the background of how this dataset is being used, but this quantity will fall over time. So what helps, on a more positive note, is if we are able to compensate for pursuing less likely hypotheses, of course, with greater power. I think a good example from human genetics is going from the candidate gene era to the GWAS era, where the first GWASs, the genome-wide association studies that were conducted, we learned about the effect size spectrum, and then we increased sample size considerably into the tens of thousands and even hundreds of thousands to find significant associations. And I think what you've seen there is lower effect sizes than initially hoped for, but tremendously reliable results with respect to what was previously done in human genetics. There's a lot of counterintuitive findings that we see. So this is looking at the studies per relationship. So the blue bars on the top are the fewest studies per relationship, all other things being equal, and the green bars are in the middle, and the red bars are the most studies per relationship. And so this was pretty counterintuitive to me at first. If there are many studies essentially being conducted by independent labs for a given relationship, we would normally think that type of corroboration is helpful for reproducibility, but what often happens in practice is that they're selectively reported, and the ones that are reported from that set tend to be less reproducible over time in our analyses. So those are summary patterns of the expectation of this quantity that we've developed, but of course there's variance around those values as well, and so each of these dots is a different realization of this stochastic process, and what we've looked at here is how liberal the data governance is, and what we mean by essentially as a proxy for that is how many studies are being conducted per data set. And so on the green here is very liberal data access policies where everyone can conduct one of these studies, but they're not overstudying a given relationship. And on the red is very restrictive data access policies. And as you might expect, this quantity itself is much more variable when we have very restrictive access policies because essentially the quantity scales with the inverse of the number of studies being conducted. And so I think counterintuitively to me, what this means is if a reproducibility audit of a shared data set is being conducted, it might make sense to conduct it on a small set of studies, but that might be the most noisy and the least reliable essentially setting to conduct that analysis. So I think, when you're going through the literature, it can feel like this. I think if you remember one thing from the talk is that these structural factors and structural context can help tremendously in understanding reproducibility. I think just in summary, shared data sets, they facilitate rapid, simultaneous testing by many teams. I think this is a multiplicity challenge that we haven't really addressed that really rivals high throughput genomics, but is handled completely differently. The multiplicity in these settings should be addressed. I think we've pitched essentially just one way to do that. There are many others. Reproducibility in a shared data set, I think, should be viewed as a stochastic process, and its variance should be appreciated. And then structural factors, really, we found can be as influential on reproducibility as individual investigator responsibility. And last, that these reproducibility audits depend on variance. So I just wanted to thank my PhD supervisor, Zach Kohani, our co-authors here, and colleagues Johnny Anidis and Chirag Patel, Zach's lab, and then the organizers for inviting me. Thank you.